Hello, my name is Dr. Roderick L. Roll, and today I will be discussing molecular bases of inheritance. So previously we discovered the genetic basis of inheritance, how chromosomes are moved through mitosis and meiosis. We've talked about that. Now we're going to look at on the molecular level, what happens, what is being transferred, what is moving. So this brings us to 4 questions to address this chapter of molecular inheritance. The first question is transfer even possible? The second question will be, what is the transforming element? The third question will be, what is the monomeric structure? And the fourth question is, what is the polymeric structure? So let's address the first one. Is transfer even possible? This takes us to an experiment by a scientist named Griffith. Griffith looked at bacteria and how the bacteria affected a mouse. The bacteria in question is Streptococcus pneumoniae. So in 1929, Griffith performed an experiment. He had 2 variations of Streptococcus pneumoniae. One was a deadly strand and one was not deadly. The deadly strand had a protective capsule, so they called it the S strand, or the smooth strand. And then the one that was not deadly, they called it the R strand, or rough. So what happens is, if Streptococcus pneumoniae does not have a capsule, it cannot be protected. Then your immune system will immediately destroy that bacteria. But if it has a smooth protective capsule, then it can be camouflaged in the body. Then your immune system does not see it and it cannot destroy it. So those are the 2 bacteria types that Griffith utilized. So let's take a closer look at his experiment. Mr. Griffith injected Streptococcus pneumoniae, the deadly strand, into a mouse, and it killed the mouse, which was expected. Then he took the non-lethal strand, the rough strand, and he injected that into the mouse. The mouse was able to build an immune response against that bacteria and immediately kill Streptococcus pneumoniae. So the rough strand was able to be destroyed. Now what Griffith did was he took the deadly virulent strand and he heated it. Let's say he boiled it for an extremely long time. And when you boil something, you denature proteins, you destroy the integrity of the entire organism. So he heated the deadly bacteria, the S form, and then he injected that heated deadly form into a living mouse, and the mouse lived. If you mix heat-killed dead S bacteria with living R bacteria, a transforming material passed between the 2, therefore making the rough Streptococcus pneumoniae turn into a deadly form. As you see in the image, the mouse died. So the rough bacteria is not supposed to kill, but it did in this image. Why? Because it picked up something that was left from the dead heat-killed bacteria. So to answer the first question, is transfer possible? Yes. Griffith proved that something transferred from the dead heat-killed S strand to the R strand bacteria, therefore making the R strand, the rough strand, become virulent. So this brings us to our second question. What is that transforming element? So now we know that something moved, but what is it? These three scientists would try to address that question for the next 14 years. So they formed countless studies to figure out what was the transforming agent in Griffith's experiment. These scientists were Avery, McCarty, and McLeod. What they did for the 14-year investigation was they mixed RNA from the heat-killed dead bacteria with the non-virulent bacteria. Then they mixed DNA from the heat-killed dead bacteria with the non-virulent bacteria. And then they mixed protein from the heat-killed dead bacteria with the non-virulent bacteria. And after 14 years, they realized that the transforming agent was DNA. Now, how you would perform this experiment is you would take the bacteria and you would destroy the bacteria with detergent. This would release all of the DNA, all of the RNA, all of the proteins, all of the sugars. It would release everything from the bacteria. Then you would centrifuge this slurry. Then the particles would be separated based on density or based on weight. So now that you've separated these different molecules, now Avery and the gang would pull out different fractions of that separated slurry. They pulled out the RNA and mixed it with the rough bacteria. Then they took out the DNA and mixed it with the rough. Then they took out the protein and mixed it with the rough. And they determined that the transforming agent, what made the rough bacteria turn into a deadly strand, was the DNA. And this was done in 1944. The next two scientists, Hershey and Chase, did a qualitative analysis or a qualitative test. This was to confirm an already existing answer. So in 1952, these scientists radioactively labeled a virus. They radioactively labeled the protein that is associated with the virus by labeling the sulfur. And then they radioactively labeled DNA of the virus by radioactively labeling a phosphorus. In the image, we have a typical bacteriophage or a phage. Notice that a virus is not a living entity. It has two major components: a protein capsule, and inside the protein capsule, you're going to have nucleic acids. The nucleic acid can be either DNA or RNA. So for this study, they used DNA. The next thing Hershey and Chase did was they allowed the phage to infect the bacteria. So the phage that the protein coat was labeled, he allowed it to infect the bacteria. Then the second parallel experiment, he allowed the phage that had the radioactive DNA to infect the bacteria. If you look at what happened in this experiment, if you look at the first one, where the protein coat was radioactively labeled, notice that the phage did its thing, but no radioactive element was found in the bacteria. If you look at the bottom image, the phage that had radioactive DNA, when it infected the bacteria, notice that when the cell was harvested, it had radioactive particles in it. After the phage was allowed to do its thing, you then would need to separate the phage from the infected bacteria. So the phage was introduced to the bacteria. It did its thing. We just don't know, is it putting protein in there or is it putting DNA in there? So you then centrifuge to separate those two components. The bacteria is heavier than the virus. So the bacteria will collect in the bottom of the tube that was centrifuged. It will be in what is called the pellet. The virus will be in the supernatants. So you can remove the virus away from the bacteria, but the virus has already done its thing. Now we have the bacteria by itself. Test the supernatant for your phage to make sure the phage is actually in the supernatant. Then test the pellet for the bacteria. The bacteria would then be lysed with detergent. Once it is lysed, you're looking for radioactive particles. In Hershey and Chase experiment, the phosphorus was seen in the bacteria that was ruptured. Therefore, this concluded that the DNA was the transforming agent, not the protein. So the second question was answered. What is the transforming element? The transforming element is DNA. Number 3, what is the monomeric structure? Each nucleotide is a monomer containing 3 parts. They all will have a phosphate group, a deoxyribose sugar, and a nitrogen containing base. Each cell contains 3 billion base pairs. The only difference between nucleotides are the nitrogen containing bases. If we look at this image, we have 2 categories: purines versus pyrimidines. The purines will include adenine and guanine. They're in that category because they have 2 rings. We call it a double ring. Now, if you look at the pyrimidines, thymine and cytosine, they only have 1 ring. So what is the monomeric structure? It is a nucleotide. So now let's address the 4th and final question. What is the polymeric structure? To address this question, we're going to talk about Chargaff. So how do we know which nucleotides pair together? The scientist named Chargaff deciphered that for us. In 1950, he performed many experiments and he saw that the adenine and the thymine had the same concentration. So therefore, they must be together. And the cytosine and guanine had the same concentration. So they must be together. So if you look at the image below, you see the red star by humans. So he did a analysis of human cells and he looked at the base composition. He saw that in the human cells, he had 29% adenines, 20% guanine, 20% cytosine, and 30% thymine. You would therefore deduce that A and the T must go together because they are the same concentration, and G and the C must go together because they are the same concentration. Let's look at the next characters in this discussion. John Randle. He was offered a professor position at King's College in London, and he asked his friend Maurice Wilkins to assist him in using physics to study biological problems. So in this image, what they're doing is they are bombarding DNA with radiation. And what is going to be obtained is an image. Then these scientists will deduce how many strands are in DNA, the spacing of DNA, and so on. So Wilkins was tasked to help use physics to understand the biological problems. So Wilkins hired Franklin. Rosalind Franklin, worked in his lab. She was tasked to use X-ray crystallography to study the three-dimensional structure of DNA. Crick was also at the facility, but he studied protein structure using X-ray crystallography. In 1953, Rosalind Franklin's X-ray crystallography image suggested that DNA molecule was a double helix of even width. This is why we currently know that the DNA polymer has two strands. It was because of this image that Rosalind Franklin obtained. She obtained this from purifying DNA. Once she purified it, she bombarded it with X-rays. The X-rays diffracted and created an image on a piece of film paper. Then she analyzed what the image suggested. Other scientists had other theories about the three-dimensional structure of DNA. Linus Pauling had a different theory, and he suggested that the DNA molecule was composed of three strands, a direct contradiction to what Rosalind Franklin thought. She thought the DNA molecule was composed of two strands. Franklin's image suggested the nitrogenous base faced the interior, while the sugar backbone faced the outside aqueous solution. So in this image, we have double-stranded DNA moving in opposite directions. I circled the nitrogenous base portion. The nitrogen-containing bases are facing the inside, and the sugar, where the red arrow is facing the outside. So Rosalind Franklin's image suggested this. Watson came to visit Crick, and Wilkins allowed the two to see Franklin's data. Wilkins, who was over the lab, allowed these two visitors to see her journal. And from looking at the journal, Crick and Watson was able to decipher what her image was saying. Franklin was not aware that a breach in protocol occurred. She did not know that they saw her images and they stole her data. Now, from this debacle, Watson and Crick subsequently won the Nobel Prize. They created a three-dimensional model of the DNA molecule in 1953. They subsequently came up with 6 discoveries while creating this three-dimensional model. The first discovery was that a DNA molecule is composed of 2 long strands of nucleotides paired together by these hydrogen bonds. Number 2, the 2 strands travel in opposite directions. Therefore, it is anti-parallel. Number 3, a full helical turn is 3.4 nanometers in length. Number 4, 10 bases make up every turn. Number 5, bases are 0.34 nanometers apart. And finally, number 6, the molecule is of uniform width. So these are the 6 discoveries that Watson and Crick came up with from looking at Rosalind's image. This uniform width suggests that a purine will pair with a pyrimidine. This further confirms the Chargaff rule.